Good day, this is Professor Will, CBMD PhD. Today's Wednesday, July 21st, 2021. It is 8:36 and 59 seconds p.m. Eastern Standard Time. Today's research, cell differentiation. So let's hop to it here. The strong signals, modeling the dynamics of cell differentiation in the development of bile ducts. Research may lead to an improved understanding of embryonic signaling pathways. Date July 20th, 2021. Source, University of Tsukuba. The scientists employed a mathematical model to simulate the differentiation of epithelial cells based on signaling molecules from the liver's portal vein. And this work may lead to new tools to better understand the very complicated signaling pathways involved in cell differentiation. The researchers created a computer model to simulate the development of complex structures based on the delta notch signaling pathway, and this work may lead to a more comprehensive picture of the process that results in the formation of organs and other physiological systems. The development of a tiny embryo consisting of undifferentiated cells into a healthy fetus with spatially defined organs depends on the complex interplay between genetic instructions and signaling molecules. For example, notch genes are found in almost all animals and insects, and encode for receptor proteins that extend through a cell's membrane. And this allows external signaling molecules to coordinate the cell's development by turning specific genes on or off at just the right time and location. However, there is still much we do not understand about the details of this mechanism. Now, to better understand the role of signaling systems in organ development and cell differentiation, researchers created a computer simulation that models a delta notch signaling pathway in biliary cell differentiation. The differentiation of epithelial cells that are essential for the development of liver's bile ducts are special in that they receive signals in the form of delta ligands from portal vein cells to ensure they are in the proper location. A delta ligand released by a portal vein cell can bind to a notch receptor to regulate gene expression in the epithelial cell. The scientists use a set of coupled differential equations to show how the concentrations of each change over time on a 20 times 22 dimensional matrix, mimicking the planar cross-section of the liver. The diffusion of delta molecules led to concentration differences based on the location, ensuring that epithelial differentiation occurred at only the correct places, which the authors called fine-grained differentiation. However, even with a portal vein cell from the liver sending out delta molecules, certain conditions resulted in no cell differentiation, showing that proper development is dependent on the rates of production of delta ligands and notch receptors. And this project demonstrates the ability of computer models to simulate the formation of spatial structure using complex feedback signaling pathways. Future models may incorporate other signaling molecules, as well as cell migration. Good day, this is Professor Will, CBMD PhD, on microbiology, bioinformatics, and pathophysiology. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.